Well, I hope I picked the right microphone, but I did. Thank you, everybody, for coming back into this big auditorium. As said yesterday as well, feel free to come to the front. The screen is big enough, but it's nice to see people being around to listen. So a very warm welcome from my side back to the ELIXIR track. There's one more session today, and we are having presentations from some of our ongoing efforts in the ELIXIR communities and also one of the focus groups. So we will start with Fotis Psammopoulos from the Institute of Applied Biosciences at the Center for Research and Technology, Hellas CERT, who is presenting work done by the ELIXIR Machine Learning Focus Group, which is co-led by himself, as well as Silvio Tosato. I'll leave all the details to him, and you can already see the title, which is the ELIXIR Machine Learning Focus Group's Achievements and the Road Ahead. Enjoy.
Thank you, Katharina. So hi, everyone. I'm actually quite excited to give you an update on that, mostly because there has been a lot of discussion about AI and machine learning these couple of days, so I think it's really fit the theme. So I like this slide mostly because it doesn't say anything beyond the obvious, that there's a lot of data. But one of the key points that I really like here is essentially that because we have all this data, it's actually possible to start thinking more effectively around what machine learning can do in this context. And this is the other favorite slide I have. I don't know how it translates in other countries, but in Greece we have this saying that we are talking about astronomical amounts of data. But actually, if you look at it, and this was always already a few years ago, biological data is actually a lot bigger than astronomical data. So it's sort of setting the perspective. So, machine learning. So we've already heard several different applications of machine learning. And I think it's quite evident at this point that it permeates life sciences. You have machine learning at the cell level, trying to figure out about cell type annotation, for example. You're going to the genome level, identifying gene regions, or doing anything about gene expression, profiles, everything else. We go to proteome. AlphaFold is the more key example here where we solve the protein folding problem to an extent. And then you have system biology, metabolism, pretty much any aspect of life science you might find an opportunity for machine learning to play a role. So that's good, right? And yeah, I think if you actually go into literature and do a kind of search, you see this kind of an exponential growth in literature. And the query itself, you can see it there. Oh, there's a pointer. You can see it there. It's not too restrictive, but it still gives a very good assessment of what is actually happening in life sciences. So everything is fine, yes, and now comes a quick question. Usually I have this as a kind of poll of sorts, but I'm not sure this will work in this context, so I'll just leave it for just a few seconds to think about it. The question is rather simple. You have this enormous number of publications. In 2020 only, you have like 9,000 publications on machine learning in life sciences. What would be the percentage of all those publications that have been accepted, are published, are part of the corpus, that have none, like zero metrics on evaluation, like no information whatsoever, from accuracy to how well it worked, anything? So that's kind of an open question. I just leave you just a few seconds to think about it. It could be anything from like 0%, that would be ideal, I guess, to 100%, this is catastrophic, no one is giving anything. So drum roll and stuff like that. And we go to this one. So this is an article that came up just a couple of years ago. And they did an assessment, and they went through the literature and said, okay, let's see, do you have any kind of an evaluation? And ultimately, they showed that roughly 20% of publications, like 1 out of 5, had absolutely no evaluation in it. Like, I did machine learning, and this is the biological insight. How? What? What about the machine learning aspects? Nothing. That's it. And interestingly, if you actually look into the co-authors of these publications, it appears that if you have absolutely no one in the co-authors list with computational background, this percentage goes up to 34%, it's like 1 out of 3. This is not good, because ultimately, this is not good science. And Just to provide a bit of what we're talking about when we're saying about evaluation. 20% of this kind of line is the green line, which is a big number. And no evaluation means, for example, you have no information about independent data sets. There might have been, or there might have not been, no information there. About cross-validation, did you have any kind of mechanism to ensure that there is a robustness in the whole process? Kind of statistical task, like what was the p-value at the very least, or something, confidence intervals, anything, any information that can assess that this actually works. So this is not good. And this actually led to a very good question. So it's not sufficient to present results. And this is one of my favorite tweets, so of course not mine. How can we deal with that? And the answer, or hopefully one of the answers, is to talk about standards. And this is where the machine learning focus group of ELIXIR comes into play. So this is, in one slide, who we are, what we're trying to do. So it is being co-led by yours truly, Silvio Tosato from ELIXIR Italy, Leila Zael Castro from ZebMed, and Jennifer Harrow up until recently before she moved to AstraZeneca. And broadly speaking, the focus group is trying to capture all aspects of machine learning for life sciences, from standards, which I'll be talking more in a few minutes, benchmarking, training, fairness, first and foremost, and ultimately how to make sure that these aspects are part of the ELIXIR community in general. And it's from the get-go, it was quite an interesting group because there were a lot of participation throughout the ELIXIR community, consistently with a lot of participation there. Brief timeline. So we formally started in 2019, so 3 years now, almost, actually to the day. And from the get-go, we had a lot of participants in there. There was a fortunate moment that if you go back to September 2019, there was a comment in Nature Reviews Molecular Cell Biology about setting the standards for machine learning in biology. And the argument in that article said that we have a problem. We don't know how to report stuff. And there is no consensus on what needs to be done, especially in life sciences. So this gave us an idea. How about, well, we are in infrastructure, we are ELIXIR, we should be able to at least do a community kind of a thing. So we started having a discussion within the group, and I'm very happy to say that basically 5 months after we formally started as a group, we're able to submit these DOM recommendations, this paper to Nature, while also being submitted to arXiv, so the wider community was able to review and have information about it. In the meantime, because review process takes time, as you all know, we were trying to engage with other communities, other groups, making sure that we can have any possible kind of input, feedback on whether this works or not. I'll go to the actual DOM recommendations in a bit, but the main idea was to provide some guidelines, some recommendations, as the name implies, on how to do effective reporting, how to make sure that whatever you read in the actual article reflects the reality of what was the machine learning. It doesn't talk anything about assessment, quality, reproducibility, anything about it. It's literally just tell me what you did. I need to know that because otherwise I cannot assess what you're doing. So the DOM recommendations, the DOM actually is an acronym, not mine, again. Other people are more creative than me on that front. And it stands for Data Optimization, Model, and Evaluation, the key components of any kind of machine learning process. It was, as I said, ultimately accepted Nature methods. And since then, we were pushing a lot in how to make sure that this is useful. It is adapted by the various communities. It's being essentially used by the ultimate users. So in the next few slides, I'm just going to go through what it actually means and what are the recommendations. But I will not go into too many details, mostly because I'm sure I'm going to bore you to death, and that is not my intention. So in every case, I'll have this kind of bullet points, and then I'll directly shift to the next slide, which actually gives some examples. So for example, when we're talking about data, I have 4 main components. And the components are about provenance, data splits, redundancy, and availability. Why are these critical? So when we're talking about machine learning, the first point is what was the data that you used to do stuff, right? And provenance is the question, all right, you selected this data, where did you find it? And why did you select this particular data set in particular? Did anyone else use it before you? What was the rationale before in using that? And also, importantly, did you use it as is? Did you get any splits? Did you have validation sets? What did you do with that? And going one step further, if you actually did those splits, was there any redundancy? Were they completely separate, or was there overlap? Because this has an impact into the actual machine learning process. And finally, yeah, you told me that this is the data, you described it, but can I actually find it? Is it somewhere accessible? So those points actually allow me to understand what was the starting point. What is the data that you applied this process in the first place? Then it goes to the optimization. So again, I'm going to skip that and go direct to an example. So I did not say that before, but this column actually are what would be possible replies on these corresponding questions based on based on this particular paper that I had here, which was selected semi-randomly, I'll put it that way. So in terms of optimization, this is how the actual machine learning process was running. So for the algorithm, the first place is, what did you actually use as the algorithm? And here the question is it a new method that was developed particularly for this one, or is something that was used in the past? And then the follow-up question in my mind is always, okay, if it's a novel method, and you actually report ultimately novel biological insights, then why did you not first submit the method itself for peer review before going to the biological insights? Because you have new method, new insights, and new process altogether, it's much harder to actually evaluate in total. We're talking about meta predictions. What is the goal here? What was the encoding used? Did you use it as is, or did you use any kind of transformation? Parameters, well, any kind of algorithm has at least one or two parameters, where there are very few that you are running as is. What are the features and how did you encode them if need be? Was there any fitting assessment done? Because you usually have kind of overfitting, underfitting. How did you assess that? And regularization. And finally, did you provide this as a configuration? Can I reuse all this setup for me to reproduce the entire thing? The model is the actual output, right? So you have the model, what is about it? And again, some of the first question is about interpretability. And this was raised very elegantly in today's keynote speak. So this is basically the same question. Is the actual model transparent? Is it actually interpretable, or is it a black box? Just to put a very slight note here, none of those points are to assess whether this is good or bad. The question, and I want to really stress this out, of the DOM recommendations is to ask, please report it. I'm not going to judge whether this is good or bad. This is not the purpose of the recommendations themselves. This is going to be up to the peers to evaluate and to assess. What I'm asking is, please let me know this information. The response might be, no, this is a black box. That's fine. From the DOM recommendations point of view, this is sufficient because it is clearly stated. The question is whether that is useful or not. This is not part of the DOM recommendations. Then we go to the output. What is the actual output of the model? What is the execution time? Again, it's quite useful for me to know that I need to run this in the cloud infrastructure of an entire country and run it for a whole month, versus I can run it in my laptop like in 15 minutes. The hardware requirements, execution time, are useful parameters to be aware of. And finally, as always, you have the data available, you have the configuration available. How about the actual software? Can I use the software and rerun the whole thing? And finally, the crux of the whole thing, when it all started, evaluation. So did you actually evaluate anything? And if so, what were the metrics that you used to do that? These are key components, because otherwise I have some strong concerns about how I can proceed from there onwards. A comparison is always a useful case. So you did the evaluation, did you compare it against something, or is it just, I'm reporting that I have a 90% accuracy, period, that's it? Did you have another data set that you tried again, another method? Was there any comparison done against the baseline? And going a bit through into statistics, were there any confidence intervals reported, or just a number like 90% accuracy, that's it, take it or leave it? And finally, again, are you making this available? Can I look into the actual raw details of the entire thing from the process? So all those are parts of the DOM recommendation. And I really urge you to go through the actual table, because it's quite useful to understand. So I want to wrap this up by sort of setting the stage of what we're trying to do now. So right now, this was one of the first outcomes, and I have to say I'm really proud of that. But we are not done yet, not by a long shot. We have a lot of short-term activities that are led with, by a lot of amazing people through the machine learning focus group in talking about how to further work on the DOM recommendations, on machine actionable recommendations, how to find about gold standard data sets and how they are defined, to talking about synthetic data, how you can assess them, what are the available information there, and so forth. But there are also some long-term activities. And the long-term are how to continue maintaining the outputs of the machine learning focus group. For the time being, the DOM recommendations, but this is going to be beyond that. Setting a governance structure so that other people can get involved from other communities is the key step in doing that. And we've already setting up, for example, a steering committee for the DOM recommendations on how to proceed. And also kind of implementation. ELIXIR is an infrastructure after all, so it needs to provide kind of a service that people can find it easier to use all those standards. And machine actionable recommendations is definitely a key point there. And I'm closing with this kind of a slide, like a vision of how we hope this would evolve. So, of course, ELIXIR it's much broader than machine learning. But at the same time, if machine learning is so pervasive at this point, it's quite interesting to see how we can further adapt the DOM recommendations, the standards, the data management aspects that exist within ELIXIR across the various relevant communities. And we already try to do that for the DOM recommendation. We have connection to NIH, to RDA, OpenAIRE, particular networks like the NFDI for data science, if I'm pronouncing it correctly. So we do have a goal of moving this forward. Ultimately, again, this is kind of a vision statement, if you like. The idea would be to have ELIXIR, the standards, again, not only DOM, but all of the standards around data in the center, connecting and adapting those to the needs of the particular communities around that. With that, I would like to acknowledge my co-chairs, and in particular the entire ELIXIR Machine Learning Focus Group, because this is, at its core, a community effort. We were mostly leading that. And thank you, and any questions, I'm more than happy to take them.
Thank you very much. Do we have any questions in the room? Well, we probably have time later on to follow up. Thanks again. Great. We're moving on to what is a very different topic, but also there's a lot of data involved. So I'm welcoming Toni Gavaldon, who is an ECCB researcher, partly working at the BSc here in Barcelona and part of INB in ELIXIR Spain. The title of the talk is the ELIXIR Biodiversity Community, Biodiversity Data Aims and Challenges, and the work represents efforts from the ELIXIR Biodiversity Community and the other co-leads, Josie Bergin and Rob Waterhouse. Thank you.
Thank you so much. So as he said, I'm co-chairing this emerging community from ELIXIR, together with Josephine Bergen and Rob Waterhouse, and the intention today is a bit explain a bit what are our intentions with this community, and also what has been done as a focus group that was previously. So I think I don't need to explain what is biodiversity, but you probably understand that it's a very massive and complex issue in which we are talking about all the individual species and variation within and between that populates the whole planet. We are talking about the variety of forms and diversity across species and also within species and between different populations. And this is a massive amount of data if you think about it. Also, in the current situation that we are now, we are in the Anthropocene, and of course, human activities are leaving a footprint on our planet. And this is impacting biodiversity in many ways, most of them being non-positive. And this impact can be through, all our activities, industry, agriculture, globalization, and all these things. And this has led to a situation that has been described as a crisis, and there is several data that points that maybe we are already starting into the sixth massive extinction event, if we count the massive extinction events that have been on planet Earth. And this was highlighted by the recent report by IPBES, the Global Assessment Report on Biodiversity and Ecosystem Services, where they highlight data that are on different organisms that show that the rate of extinction of the different organisms is really increasing since since then we have some measures. So this kind of parallels similar data regarding the climate change crisis, and we should be embracing this as also a very important challenge. And the thing is that we still don't know all this diversity, and probably the diversity is being lost at a rate that is faster than we are discovering it. This is just a tree of life, just to highlight that most of the organisms we study are multicellular, large organisms like animals, plants, and some fungi, but this constitutes a small corner, and most of the diversity is encompassed by microbial groups for which we still know very little. Even if the massive amount of data we have now, almost every year, new clades are being discovered by efforts for sampling, environmental DNA and so on. So in all this process of knowing the diversity and studying that and discovering, classifying, it seems that, it's clear that molecular data is key for all these processes. We need molecular data now, and it's being used to discover biodiversity. So all these metagenomics projects that are sampling, sequencing samples from all over the planet are revealing lots of new species and new organisms that we are not able to cultivate and that we never seen before, and that it's showing us that the diversity spans a magnitude that we were not expecting before. We need also molecular data to classify, because it's been proven to be much better than looking at morphological characters or other things. And also we need molecular data to understand this biodiversity, to understand the evolution, to understand how different organisms relate to each other, how they may have originated, and how their features may be explained by the changes in their sequence. And it also has been shown to be very useful to monitor biodiversity, to monitor whether biodiversity is changing from year to year, and this is a good way in which we can measure what is the impact of whatever human activity. So, and molecular data, we have been talking about this a lot in this meeting, is massive. We need to be processed, analyzed, integrate, etc., etc. But regarding biodiversity, when we talk about biodiversity data, we are not just referring to molecular sequences. We are also referring to a lot of data that is really important to understand biodiversity. And this is the knowledge graph for biodiversity by page, in which it relates different types of information that we would like to have attached to each sequence, like to know where that sequence was sampled, like GPS data, to know the traits of that organisms to which the sequence belong, etc., and to have maybe images, to have additional information that we could relate to. All these is different types of data that we have to integrate, and that also adds to the magnitude of data that we need to analyze. So in this context is where this focus group was set up. So it's clear that there are societal challenges impacting biodiversity. There is a growing recognition of the threat that we are putting into biodiversity and the danger of fast decline of this biodiversity. We don't even know what is out there. We don't even know what is the impact of our activities, in what way we are moving, and we still miss solutions, and the policymakers need information and accurate information to have the decisions. As I said, there is a lot of informatics challenges for biodiversity data, the need for integration, the magnitude of the data, the need to move data and to consult data from different parts of the world and integrate all these. And there is also, we see opportunities in this context. First of all, because biodiversity is usually studied from discipline perspective. So people interested in amphibians, they study amphibians, and there may be a large community of amphibians well connected. Then there's other people focusing on a given group of plants or some on microbes. And there is a big opportunity, I think, for ELIXIR as a hub to connect all these different communities, because actually they have the same needs in terms of data analysis and integration of different types of data. So we see these scientific opportunities in biodiversity, like we see in the future. We think that sequence-based biodiversity monitoring will be the default for studying like impact of human activities and so on, because it can be done faster than observations and so on. We envision also that in the future informatics will connect molecular data with other types of data, and that knowledge will be the basis of applied data-driven science and also for informing decisions. And we also see that there is a big opportunity for empowering citizens to monitor also biodiversity, because they are living everywhere in the world, and if we put some of these monitoring technologies and opportunities in their hands, we can multiply our ability to monitor what is happening with biodiversity. So all these are opportunities that we think ELIXIR can contribute to. And this started, in 2019, started this first as a focus group. And this is a bit the story of the group. There was a meeting face-to-face in Milan. There has been a lot of virtual meetings. There's regular people attending from 8 to 9 nodes of ELIXIR. And there was a big effort on an opinion recommendation paper that was published last year in Faculty of Thousand Research. So this is the paper, and where from the discussions of the focus group, there were some recommendations that are listed here. So as you can see, that there is a need of collaboration between biodiversity-related and molecular-focused infrastructure, which are usually separated and disconnected. And there was a realization that the taxonomies need to be aligned and harmonized across domains. So the taxonomy of bacteria has nothing to do with taxonomy of vertebrates, or not even with other groups. The approaches for managing molecular data, there's a need to scale up, because the scale is just growing exponentially. There's need to be for training, and community needs to seek common solutions that enable molecular technologies to advance biodiversity research. So this is basically the core messages from this paper. We also see that we are a community within ELIXIR, but we do see a lot of connections with other ELIXIR platforms, communities, or focus groups. And here we highlight those. I mentioned already the need for training, computing, interoperability, data, and also platforms like Galaxy could be important also to bring these technologies to more widespread users. Of course, the groups focused on plants, or the groups focused on marine metagenomics, where many molecular data is being available from a lot of hidden diversity. So we do see all these connections, and this is one thing we want to collaborate. So this is a bit the idea we have for the immediate future. Like, we are transitioning now from a focus group to an ELIXIR community, and we want to grow the network, to start increasing the contacts with other ELIXIR nodes, and also to increase the portfolio of ELIXIR services that could be relevant for biodiversity. There was an initial screening of which of those services could be important for biodiversity. We want to increase this portfolio. And maybe ELIXIR can be a partner for European projects, or can help to organize people around to go for European projects, and so on. So also leverage ELIXIR platforms to support biodiversity research, maybe adapting some of them or some of those. And that's basically what I wanted to say. If you are interested for joining us, you can join a mailing list, or you can contact me or Jerry. And yeah, also we have monthly meetings, so I'm welcome some questions if you have some. Thank you.
Thank you very much. Yeah, I think this is a nice example how efforts come together and then are being moved forward. Do we have any questions in the room?
Yeah. Hi, Tony. Very nice. So if I understand it correctly, you are talking more about annotation of biodiversity and standardization. And my question is more about all these projects that are around now to sequence vertebrates, the tree of life. So whether there is any kind of connections with that too. And then I have a second question, so maybe you can ask.
Okay, so yes, this is, I didn't mention it specifically, but one of these efforts are, sequencing all these organisms, like the erga and all these. So all of these will bring needs to store the data, to attach other types of data. So we do see, some of us are part also of this consortia, and we do see the needs. An important thing is this consortia, they may be different because one is focused on birds, the other one is focused on other organisms, but maybe the needs, the bioinformatics needs are the same for all of them. So I think ELIXIR there has a chance to collaborate, yeah.
Okay, and my other question is about any efforts of what are you thinking about all these microbial dark matter components? Because I have the feeling that the more samples you sequence, the more unknown things you find, and it feels like there is no limit. And I think that there is a large field there to provide solutions. So whether you have also thought about that.
Yeah, we have discussed. This is like a working group that we discuss the challenges that are ahead, and this, of course, relates to this. There is massive amount of data being produced. This that needs to be organized, needs to be classified somehow. This needs to be translated into what is the diversity, and also it's important to monitor how this may be changing. We don't know how many species are disappearing. Maybe these metagenomics projects help us. If you sample the same time again and then you count how many sequences you don't see anymore. So that's maybe a way to measure the broad impact of the thing. So yeah, I think this is truly, truly relevant. Yeah.
Thank you.
We have one more in the back.
Yeah, thank you very much, Tony. It's great to finally see a talk about actually biodiversity. Sorry, my tone is really weird through this microphone, I just realized. Anyways, my question is, following up on the first question right now, obviously we also know that conservation of endangered species depends on intraspecific genetic diversity, genomic erosion, and so on. So are there any plans to also support intraspecific population genomic projects and also evolutionary genomic projects, I guess?
Yes. Our concept of biodiversity is not only definition of a species and differences between species, but also diversity within each of the species, understanding how the genetic diversity in different populations change. So we consider all this within the concept of biodiversity, because indeed is biodiversity. So yes. Also, I want to say that this is, is not a group that we want to solve all the issues, but it's kind of a space in which people can come and discuss their issues and find partners with similar issues, and then ELIXIR can maybe mobilize to solve a specific problem. So yes, we welcome people interested in this type of issues. Yes.
Thank you.
Thank you, Tony, and I think you just said it. Yes, if there is anyone in the audience interested in the topic, do reach out to us while we're here or get in touch by email. It is a community effort, and we can only move on if we do this together. The third speaker of the session is Jordi Rambla. He works at the CRG, is also part of the Spanish ELIXIR node. And I'm not going to say very much because I'm sure that he will cover all the details. He'll be talking about federated EGA. I think there's a couple of updates that he can be sharing today, and we look forward to your presentation.
Okay, thank you, Katarina. So good morning, everyone, and thank you for attending this talk. The talk is touching a little bit on the human data communities and a little bit more on the federated EGA itself. So the human data communities nowadays for ELIXIR are these three ones with the round logos on them. We have the federated human data that I will explain a little bit better later. Human copy number variation that it's, as you can imagine, quite focused on something. Rare diseases that Nuria yesterday give some insights on it. And originally it was also another community that it's called Beacon, that now it's kind of transversal to all of them and having the same activities, but not really listed as a community anymore. But many of you could wonder if this is still something on it. The human copy number variation goals are the ones that we are seeing here. It's led by Michael Baudis and a couple more colleagues, Christophe and David Salgado from France. They work around the copy number things. So the standards to define it, how to detect them, which are the optimal pipelines, and all these kind of things that you can imagine that are quite interesting and still quite complex. And I don't know if this will go better or not in the future because it's a complex topic itself. Then we have the rare disease community that it's focusing on providing services, solutions, and talking and discussing and making the rare diseases disease domain more advanced in Europe. And we have Serena, that is the lead of the human transnational, and I don't remember the name, HGTD, that it's the human genomics and transnational data or something like that. We have Ivo Good from the SYNAX CRG and also Sergi Beltran from it, and Marco Roos that's from the Netherlands. Note that it's one person that you will identify as a lead on the FAIR topics, and Nuria is part of his team. So the rare disease community is based also on the outcomes of the federated human data, and in particular related to the EGA project as the provider of the raw storage for the data and some solutions for it. So the federated human data community, was originally called human data community, but then we had the human data communities and the human data community, and this creates lots of nightmares and confusions and collisions in what we should talk about and what this meeting is about. So we changed the name to federated human data, so it's easier to understand which one is which. But basically is discussing about managing sensitive human data in this current context that has been described before today and probably yesterday that I couldn't attend. But we need more data. One example is in the keynote this morning about that some data sets are too small to drive conclusions is something important. Also, we had mentioned about federating stuff, creating networks of things. So the federated human data is about that. It's about creating networks or network of human data, sensitive human data that could be analyzed kind of together and at least with easier than going to every repository and trying to get the data from there and starting from scratch in every case. So this is an ecosystem of interoperable services that allows this analysis at population scale, and this means big population, both biomolecular data and, of course, what we call metadata, but I hate more and more calling that metadata. So for me, this is phenoclinic data. You can do analysis on them on its own, and you don't need molecular data for doing that, and it's not metadata for the molecular data. It's data on itself. So what is metadata is who collected that, when it was collected, how it was collected, but not data properly. So I vote here to reduce the use of the metadata name to something that is more concrete. Okay, you can see here that the services are focused on secure submission, so sending the data, as we said in the case of COVID-19 also this morning in the previous session, that this is an issue, sharing the data and submitting the data. In this case, should be secure indeed, because it's sensitive data in most of the cases. How to archive this data, how to distribute the data when this is authorized or should go to a place where it should be analyzed. Okay, so all these kind of different steps is what we are trying to tackle a little bit in this. I would say more than tackle, is to coordinate in this community. And the community is led by Giselle from the Hub, Thomas Keane from the EBI, it's my colleague at the EGA, Dylan Spalding, that now it's at CSE, but also was at the EGA before at EBI, and Giselle also was at the EGA, so you see the motto here, and myself. Okay, so how we achieve the vision, the one that I described. So we tried to coordinate, as I said. As you can imagine, the human data was one of the first communities that ELIXIR had. Why? Because a big push when you go to your, the ELIXIR is based on nodes, as you know. So nodes are roughly equivalent to countries. And then for the funding, you need to go to the ministries and you ask them for money. And most of them say, okay, how I will translate that into something that my population, my citizens will appreciate. And the first answer is always how this relates to health, how this relates to translate science from science to bedside, and so on. So the human data and sensitive human data was almost the first one in the list of data that should be, or communities that should be created. So as I'm saying that we are trying to coordinate because this is one of the domains where more funding exists. In the private funding, so there are lots of projects, and most of them have similar problems, and having one like ELIXIR saying, oh, this is how you need to do the things, because we say that, this is our approach, and you should follow it, this is not. So what we decide is to go into the way of saying, how do you solve this problem? Or do you have this problem? How do you solve it? Do we have anything in common? There is existing standards outside that we can use for that, or we can push for creating standards in the context of, for example, the Global Alliance or something like that. So this is the kind of things we are doing in this community. Okay, and one of the first things that was part of the ELIXIR catalog of services and so was the EGA. EGA was kind of the new instance, the second instance of the EGA. EGA started in 2008 at the EBI and was run from the EBI, by the EBI for 5 years until we, the CRG, where I work, joined, and then it became a co-managed project. So this was kind of the second incarnation of the EGA, and now it's coming the third incarnation. So the second incarnation of the EGA was 2013, so it was kind of at the same time that ELIXIR was starting to be organized and created and funded, and then it happily happens that these two things happen kind of together. So EGA was one of the first examples where 2 countries or 2 nodes, meaning the EBI, that is one of the ELIXIR nodes, and Spain, that is another of the ELIXIR nodes, collaborate together in providing a service for the whole Europe, the whole ELIXIR, and the world wide. The whole world, sorry, this is too difficult for a Spanish guy. Yeah. So we created this archive, co-managed, and we ran this for some time. Then we start to say, okay, we have other nodes, and in 2016, here in Barcelona, in another project, meeting of a ELIXIR meeting that is a project called Accelerate, one of the tasks there was to create something that allows easier submission to the EGA, something similar to the brokering or the data hub that has been described this morning in the COVID-19 examples. But this, when we start asking the interested nodes in that what they really need, it becomes quite clear that what is required is something like a network of archives, not just people having some tools that they can deploy, like a Galaxy instance, for example, at home, and they use it, and it's a silo or something like that, or just for that country. It was clear that this is something bigger that needs to be a part of a bigger and more ambitious view. And this more ambitious view ended up being the federated EGA. So the federated EGA is, as I said, a network of nodes that share some things like the user interfaces, the protocols, the procedures. They share the identifiers. Of course, they are different, but they share the same model of identifier and many things. And after 6 years already of working in that project, now we have something like this, that is the process to become a federated EGA node in production. And you have here 2 paths. One is the let's say, more legal governance, contract agreements path, and then the technical path in yellow. So we have plenty of nodes that started just testing that they could have the technical side on the setup and configured and running without any real data, just with some synthetic data, maybe in some small virtual machines, something like that. But they have something to demonstrate to their funders mostly that this could be done, that this could be interesting, that it's not so complicated, and technically speaking, I mean. And then they start the other process that, in this case, is presenting to the federated EGA, federated human data community, which are their needs, what are their plans, what they want to do, why they need to do this, which is, as I said, the plan, and so on. And then they start to be involved in the committees, the different working groups we have to take decisions on the federated EGA, and eventually, after everything matched together, meaning that the technical is ready and all the legal parts are ready, and they have been contributing to the committees, we have an end test where we said, okay, now everything is running, everything is set up, you can go into production with this thing. So these paths are described in this website. In this website, you will find all the information. You have the link here, all the information about how to become or what this means actually being a federated EGA node, the documentation, the different things. It's not that much complicated, but when you have different people, different needs, different countries together, you need to find the way that makes life viable for everyone, yeah? and not rough. So the first step usually is that one, is presenting your use case to the federated human data community so everyone knows about what are you aiming to do and this stuff. This has been done with many different ones. We have some of them listed here. You can see that some of them indeed are not European, so it's not just for Europe. And this is the first step, as I said. And this is the current status. So we had this idea. We have this path. This path was created in this group. We have these other projects and nodes or potential nodes presenting in the federated EGA, and this is the current situation. This is the Europe map, as you can see, and we have now 4 legal nodes that are there. This is Norway and Sweden. We have Finland that signed the agreement last week. We have Germany, and you see Spain in yellow because it's getting hotter, and hotter. And I hope that we will burn our hands very soon, yeah, with this thing. So it seems so. Every news I'm hearing are positive in this way. So it's approved, let's say, but it requires the final signature of the thing. And then you have lots of other countries that have shown interest. Many of them already have done the technical test and so on. And then we have indeed other countries and other places that are asking about being part of this federation. Of course, all of them can maintain their own image, especially, for example, in Canada. They don't want to be something that says European node, and they say, well, this doesn't make sense for us. But yes, of course, Australia is the same thing and so on, but the concept is still there, yeah? And, sorry. We have also what we call the maturity model. The maturity model is something that is very, very popular now in ELIXIR. We have maturity models everywhere. I'm going to start suggesting to have a maturity models on maturity models, so we can assess if a maturity model is mature enough to be published. Of course, should be fair maturity model. And sorry, I can avoid that. And this is developed in the context of a Converge project that has been also mentioned and has been led by the BSc team and has been the contribution of different nodes also, and in the context of, I said here, different projects. And this is a self-assessment thing. So you see, I don't remember the exact number, but I think there is 60 different indicators, let's call it, and there you can see for every aspect, you have them kind of listed here, in the legal part, in the governance part, in the technical part, all these kinds of things, where you can assess where are you. So this is not for us to fiscalize if you are ready or not ready. It's for yourself to know if it makes sense to move forward or you are too far away or to get started. Remember what I said, that we started the project more strictly in 2016. The technical side has been ready almost 1 year after, and we had time to have almost 2 more iterations of the technical side until the first legal agreements has arrived. sign it, I mean. Yeah? So the path could be very different, and this maturity model is helping you in assessing where are you and not missing any important point in the trip. Okay, we have several committees. Some of them are technical, and the technical ones are mostly proposing to use global alliance standards to connect these different nodes together. And this is something that it's natural. If you want to federate something, usually you rely on standards to avoid issues. This is the technical part, so I commented that already, and this is the acknowledgements. You can see here that, for example, different nodes that are involved, and one of them, that it's Germany, has its own logo and its own branding and so on, and this is perfectly fine, and so on, okay? So thank you very much, and Kata is coming here for questions probably.
Thank you very much. I think there were lots of interesting topics. Yeah. So do we have any questions in the room?
So tomorrow we have a celebration, official celebration of the federated TGA. So we started like something leveraging that most of the nodes are here to say, okay, let's meet together and let's celebrate with a copa of cava, a glass of cava. But it's started to become a little bit bigger. So the registration is closed, sorry. But tomorrow we have the official. So maybe in the news you will also see some news about the federated TGA being presented or celebrated or something like that. Yeah?
So
yeah, go ahead.
No, using the time, maybe one question from my side. So if there is someone in the audience who would like to be engaging, yeah, what's your recommendation?
Now we created this web page in order for people to be able to understand which is the overall scenario. So because we had some institutions coming and saying we want to set up a federated TGA, and when they describe what they are trying to do is not actually what the federated TGA node is. Yeah, so they need to understand that first. So this is one place where we can start understanding what the thing is about. And there is this path, then the first step is contacting us, and then we bring you to the federated human data community to explain your use case and your needs and to introduce yourself and get engaged with the community. So this is the first steps, yeah, easy ones, actually.
Great, and I can also second that there are very easy steps to take, not just with regards to federated DGA, but in general across ELIXIR communities. So from my side, I don't see anybody who stood up and is curious. They will all come find our speakers later on. So thanks again to all the three that presented in the last hour. Big applause. And for everybody else, it's lunchtime. We hope to have you back for other sessions later on. Enjoy the rest of today's ECCB day.